Hello, everybody, and welcome. Thank you for joining us today. My name is Sandy Regler, and I am an epilepsy educator with Epilepsy Ottawa. So glad you could be here today for our webinar. We are very excited. It's entitled Genetics and Epilepsy, How Genes Contribute to Seizures and Possible Future Treatments. We are very pleased to have join us today Dr. James Eubanks, who is a senior scientist and a research division head at the Krembil Research Institute. He is also a professor in the Division of Neurosurgery at the University of Toronto. He received his PhD in physiology and pharmacology from the University of California and further completed 2 postdoctoral fellowships in molecular neurobiology, first from Duke University and the second from the University of Toronto. His research interests include the influence of epigenetic factors on neuronal function, with a particular emphasis on Rett syndrome. He is on the advisory board for both the International Rett Syndrome Foundation and the Ontario Rett Syndrome Association. In 2012, he received the Ontario Rett Syndrome Association's Award of Merit for his dedication to studying Rett syndrome. After Dr. Eubanks gives his presentations, we will have some time for some questions. Please, if you're writing questions, you can write them in the comments section and we will do our best to answer them. I would also like to mention that since this is a public forum, to please keep your questions general in nature and do not divulge any personal information. And so with that, I'd like to welcome James.
Thank you, Sandy, for the kind introduction, and thanks to Epilepsy Ottawa for the invitation to give a little presentation today. I think we can start the screen share.
Sure.
I think I may ask you to resend me that permission. Okay, so welcome everyone. I want to just start by encouraging you guys to have questions raised. If you've got a couple of places during the presentation where there's logical stop points. My goal is to try to present some of the overviews towards genetics and epilepsies, talk a little bit about some experimental treatments that are in the works, still at the experimental stage, but at least in a lot of arenas showing considerable promise, and answer your questions. I know probably many of you have questions that may be somewhat related or distantly related to the topics that I'll cover, but at the end, if you have other questions that were not addressed, please feel free to send them in and we'll do our best to answer them. So to begin, let me just do a little bit of highlighting that in cases of epilepsy, there's roughly a third of the clinical cases that have a very clear reason, reasons such as tumors, trauma, stroke, infection, autoimmune conditions, things like that, where a clinician using available screening, imaging, can very readily identify what promoted the epilepsy condition. Now, that's not true for about almost 70% of the clinical cases. And you may have heard of these referred to as idiopathic epilepsies, which simply refers to without a known cause. And that is reflected in this slide, where in the old nomenclatures, you can again see that about 70% of the epilepsies were classified as idiopathic. Now, this was about 40, 45 years ago. Now, if you fast forward to only about 5 or 6 years ago, you see that this group that was considered idiopathic has indeed changed. And what's beginning to be appreciated is what was previously idiopathic in a large way now is being identified as having genetic overtones. Now, in some cases, a genetic mutation itself, all of its own, can be sufficient to cause the epilepsy syndrome. In other cases, it's a little more complicated and requires perhaps more than one event. And so I want to go through some of those different events that lead to what previously had been considered idiopathic and is now becoming more and more recognized to be of genetic origin. So we do know that mutations, even within a single gene, can cause epilepsies. And you probably have heard of some of these. Probably the most common are the channelopathies, and that's a class of epilepsy syndromes where the gene that's affected by the mutation encodes some form of an ion channel, be it ligand gated or voltage gated. Almost 3/4 of the known monogenetic or single causing epilepsy genes relate to these types of channelopathies. What's being appreciated more and more is that what's yellow and purple on this slide don't constitute everything. There's another category that's growing in size, which represents mutations that are being identified in genes that don't fall into the channelopathy conditions. And so these would be the other genetic causes. And as I mentioned, these are becoming more and more appreciated. So does this mean that all of these epilepsies reside in one's genes? And questions, obviously, that come from that generality is that how would one's individual genetic pattern then contribute to their epilepsy? Can epilepsies run in a family and be inherited? Does epilepsy arise due to spontaneous or de novo genetic changes? And the answer to each of these questions is arguably yes, but there is a little bit of an asterisk, and that asterisk is that it's not quite as simple as a simple yes. We know with certainty that your genes certainly do play a role in what physical traits and behavioral tendencies you will develop, but very few of those behavioral traits, behavioral tendencies, physical traits, it's rarely resulting from just a single gene. It's how the different genes, as well as your own life experiences and your environmental exposures, how all of these factors play together that set what these eventual patterns will be. And that's certainly true for epilepsies. So to begin getting into this in a little bit more detail, I wanted to give a little bit of a background on what are the big players in genetics? What are we talking about, and how do some of these things arise and occur? And so the first entity we need to become familiar with is, of course, the DNA, or deoxyribonucleic acid. DNA is simply the linear strand of the bases or the nucleic acids that are present in the nucleus of your cells that contain your genetic information. The sequence at which these bases are laid down determines the code that eventually will turn into the proteins that execute the functions within your cells. There are 4 different bases that constitute DNA. We'll refer to them just as A, C, G, T in alphabetical abbreviations, but each is different, and it's the pattern of the A's, the C's, and the G's and T's in this strand of DNA that's important for conveying the genetic material. Now, as I'm sure most people will have recognized, DNA is double-stranded. It's the double-stranded helix that you've probably heard of, and it forms this type of a structure. And the genes can be on either strand going in anti-parallel directions. So you could have what encodes for a gene running this way or on the other strand. And it doesn't matter. It's the sequence of the bases in their organizational structure that matters. The base pairs will always be seen with a T base pairing with an A base and a G base pairing with a C base. And so that's what's shown in this slide. You see the blue rung is for a C. It's next to a green rung, which is a G. And so that's the tolerated bases. And as you'll see shortly, if this complementary pairing doesn't occur properly, then it confuses the DNA and it confuses the cell, and it causes something to happen that tries to correct what it sees as an imbalance. And that imbalance can actually lead to some of the mutations that cause the eventual conditions. Within your genome, you have about 3 billion of these bases if you laid the genome end to end. Within that 3 billion bases, there's about 2% of that physical amount of bases actually represent encoded genes. So only a small percentage of the total DNA within your nucleus actually constitutes something that is considered a gene. The other 98% serves other purposes, some of which we don't actually even fully understand at this time. Now, you've probably seen or heard of these sorts of chromosomes depicted as here. This is a schematic of what a human chromosome spread would represent if you looked at it under a microscope. You have chromosomes 1 through 22. These are called the autosomes. Everybody has them. This is the size proportional to their number. It's inversely proportional to their number, chromosome 1 being the largest, chromosome 22 being the smallest, sort of 21 is the smallest, 22 is close in size. But we also have the sex chromosomes. 2 X's, female, and males have a Y chromosome. And so you could argue there's either 22 autosomes plus sex chromosomes, or there's 23 chromosomes with males having an extra Y and females obviously not containing a Y. Now, it's also worth mentioning that there is a little bit of DNA that's encoded within your mitochondria, the mitochondria being a distinct organelle within the cells, and 27 genes are found in mitochondrial DNA. All of those 27 genes are inherited from your mother. You don't get any mitochondrial DNA from your father. It all comes from your mom. But all of the nuclear DNA that's seen in these chromosomes, 1/2 or 1 copy of the 2 comes from your mother, and the other copy comes from your father. So you're equally represented in chromosomal material for what you inherit from your mother and your father on all of these chromosomes that reside within the nucleus. As I mentioned, only about 2% of your genome encodes for genes, and which make gene products. And the gene products, of course, are the proteins. That means the majority of the DNA in your cell doesn't actually code for anything that exerts a function, per se, outside of the nucleus within the cell. Now, that doesn't mean it isn't doing something. It just means that it isn't something that is encoding for the proteins that will ultimately be expressed. And we'll talk a little bit about some of the possible uses or usefulness of that other 98% of the genome in just a couple of minutes. If you take a look, though, at how many genes are encoded within the genome, you have about 20,000 different genes. Now, is that a lot? Is that a little? And does it actually really make that much difference? As a reference, if you look at the genome of a worm, it has about 18,000 genes. So not a lot of difference between the 20,000 we see in humans and the 18,000 we see in worms. So unless those 2,000 different are really, really critical for human evolution, it's not just simply the genes. The more you have, the more evolutionarily advanced the species will be. And that's clearly recognized when you look at a mouse, which has more genes than a human. In fact, a mouse has about 1/3 more genes in their genome than a human does. So it's not necessarily the number that's critical. It's the structure, how they're expressed, how they're regulated, when they come on, and for what they encode at a given time and in a given place. Okay. So I always like to think of the world before COVID, back when we had fun. And if we're ever once again allowed to go out, maybe go to the pub or go to a cocktail party or something and actually interact with people again, sometimes it's nice to have some discussion topics that you can use to start up conversations. And this is where DNA can form a great platform for just introducing yourself and saying, hey, you want to know some neat facts about your DNA? Well, As I mentioned, your genome contains about 3 billion bases, and each of those 3 billion bases is about 0.3 nanometers apart. So a billion bases is 10 to the ninth, 3 times 10 to the ninth, divided by 0.3 times 10 to the minus ninth, means every cell of your body has about 1 meter of DNA that's crammed into it. Now, if you look at just your brain has about 200 billion cells, neurons and glial cells. So if you took all of the DNA that's present just in your head, just in the brain, and you stretched it end to end, it would span about 200 million kilometers. That is about the same distance as taking a trip from Earth to the planet Mercury and back. And that's just your head. Now, if you then look at your whole body has somewhere around 50 trillion cells, give or take. If you did the same thing and you stretched all of the DNA in your body, it would span about 50 billion kilometers. And that's the distance that if you took a trip from Earth, it would go to planet Neptune and back 5 times. That's a great way to get a conversation started, I can guarantee you. Now, if we go back to talking a little bit more about how events can influence our DNA and influence how things function in the body, as I mentioned before, except for just a small amount of mitochondrial DNA, again, all of which comes from your mother, about half of our DNA comes from mom and half comes from dad. Now, does that mean that the sequence of DNA that you received from one of your parents will be the same as the sequence received from the other? And the answer to that is no. In fact, there can be quite significant differences in the sequences you inherit or obtain from your mother versus your father. And just a simple visual representation of that would be if your mother had green eyes and your father had brown eyes. Genes are what's responsible for encoding those eye colors, and clearly the sequence of the DNA with your father versus your mother would be different, or they would have the same eye color. And so you would inherit those traits from your mother and your father, and it would be the combination of what you inherited from your mother and father that would dictate your eventual eye color. But there's other things that can come into play as well. And this is where we start talking about some of the reasons why genetics can influence epilepsy conditions. Because in addition to just your parents having different DNA sequences to begin with, there's also random spontaneous DNA polymorphisms or mutations that can occur in the germ cells. So this would be prior to fertilization, but not present throughout the entire body of either your mother or father. And so these are just spontaneous changes. You probably have heard them called de novo alterations, and they just happen. And in fact, it's just a normal process. It's a normal thing that happens in the DNA as we're going through generation of the sex gametes. Now, there's also differences in X chromosome inactivation, and this would be in play only for females, but X chromosome inactivation can have profound effects on whether something inherited from mother or inherited from father in a woman are going to be expressed and functional. And we'll talk a little bit more about that. And then third is this relatively new recognition that the epigenetics, or the environmental influences, are also key, not to determining necessarily the sequence of the DNA, but to determining how the DNA will be structured, how parts of the genome will, or perhaps will, will not be expressed at any given time or in response to specific cueing. And so the things that are shown here in the bold and dark print are areas that I will touch on in more detail tonight. Areas lower in the regular font, I'm not going to have time to discuss, but it's worth recognizing that we can also have individual changes or individuality that's based on other factors, things like jumping genes. You may have heard of this. This is areas in the genome that can get up and move around, particularly during early embryogenesis. And as the name suggests, the gene just gets up and it moves. Well, that's fine, except where it resettles is unique from person to person. And if it interrupts something else in that genomic sequence that's in a unique way, you can also see how an individual barcode could emerge in the genomic DNA from individual to individual simply because of where those jumping genes land. And they just don't land in the same place in all individuals or even in all cells. Another thing is genetic recombination, and this is where the traits that are present in your grandparents can get exchanged with one another before they are passed on to you. And so that can also change what's represented in your mother and father with respect to, again, these sex cells prior to fertilization. And then lastly, is somatic mosaicism, and this would be things that are just spontaneously occurring in cells. And we will talk a little bit about the processes through which this can occur, but it's not something that I'll have time to deal in how it would affect the individuality of a person. But do recognize that each of these processes will contribute to your own individuality, and in some cases, they can actually be sufficient to cause genetic conditions. So let me start with spontaneous DNA polymorphisms and mutations. And so it's important for me to first define what the difference is between a polymorphism and a mutation. A polymorphism is just a change. And when used in the context of genetics, it means a change in the genetic sequence. It means one of those bases got modified. Now that doesn't make it a mutation. That just makes it a change. And we know that there are multiple changes that really don't affect negatively or positively how a system functions in the body. We all have hundreds, thousands, perhaps tens of thousands of these DNA polymorphisms, and most of them occur within that 98% of the genomic DNA that I mentioned does not contain the sequence for actual genes. So most of our genetic changes are in that 98%. Now, there's also polymorphisms that do occur within an actual gene, and they can change the encoded protein, but it's only a mutation if the change that occurs changes or alters, and almost always in a negative manner, the function of that encoded protein. So what I'll be a little, if you will, loosey-goosey with talking about polymorphisms and mutations, and I'll probably use the term mutations, but not everything that you would see, for example, on one of your genetic ancestry.com or 23andMe. Not all of those changes would be mutations. In fact, most would actually fall into the category of polymorphisms. They're still unique to you. They still define your individuality, but not all of those would be necessarily associated with something that could be called a mutation. Okay, so having probably spent too much time covering that, let's go on and do a little bit of biology. I'm a professor at U of T, and so I like to talk about how things happen. And one way genetic changes can occur is through the repair of DNA damage. And what is shown here is South Beach in Miami during good times, you know, back when people could actually go out and socialize, enjoy the sun. The only trouble with enjoying the sun is that you may be enjoying it, but it's also doing some stuff to you that you may not be aware of or you may not fully appreciate. And one of the things that it's doing is bombarding your skin, more so the exposed skin, but still even the more scantily clad areas here are exposed to this high intensity sunlight, which does contain some pretty powerful ionizing radiations. When the body is hit with this, the cells will absorb that energy, and that can change the sequence of the DNA. And I'll show you schematically how that occurs in a moment. Mostly, it hits the cells in a random manner. Most of the time when this would happen, it would occur in that 98% of the genome that doesn't encode for anything, and you'd never know it. But occasionally, there are absorptions of the sunlight that do hit an area of the genome that encodes for genes. And in some genes, there's actually hotspots, and that's probably because of how the genomic DNA is structured within the nucleus. And they're very prone to being altered by ionizing radiation. These things can cause random errors of DNA replication. And we also know that there are other things in addition to sunlight. I'm focusing on this one to some extent because of its simplicity. But we also know that things like environmental chemicals, exposure to some carcinogens or non-carcinogens, different chemicals, all of these things can also affect the DNA. And in most cases, your DNA repair systems are more than capable of fixing these problems before any alterations are seen. But in some cases, the DNA repair system actually is what's responsible for introducing the changes. And so that's what I want to go through here, just a little bit, just quickly, because it illustrates at the molecular level how some of these de novo mutations are known to arise. And so if you look, here is just the chemical structure for one of those bases that you would see in your genomic DNA. This is a cytosine, or this is a C. And the C form exists in two flavors. It can either be by itself, or it can have this modification to it. And this is a methylation or a modification that's present in a lot of the cytosines in the genome. So this is not an unusual form in which this C base would exist. Now, why is that important? It's important because this C looks an awful lot like this T. The difference here being in red, you've got this NH2 or an amine group. And on the thymidine or the T base, you have this O or keto group. What happens when this base absorbs ultraviolet radiation or high intensity light is a deamination reaction can occur. And that actually just physically pops off the amine group and it leaves a deaminated cytosine. And the cell doesn't know what is this. It doesn't look quite like a C and it doesn't look quite like a T. But remember those base pairs that I mentioned earlier where the A's bind to the C's and the C's, the A's bind to the T's and the C's bind to the G's? This would be recognized as a mismatch. And so the DNA repair systems would come back and try to correct it to restore the base that it thinks should be there. And unfortunately, what often happens is that this deaminated base gets converted back into a thymidine or a T base. So what you started with was a C, and what you wound up with is a T. And why is that important? The genetic code is based on series of triplet segments. G or CGA, for example, would encode for one amino acid that would be seen in a protein. TGA is a very different combination, and it would introduce a completely different signal for the cell in its coding. In fact, TGA is a stop codon that doesn't encode for anything. And so what winds up happening as a consequence of these exposures is that you change the genetic code, and in the most extreme condition, the one base alteration is sufficient to stop the protein from being made. And that would be the origin of some of the de novo mutations that can occur very early on in embryogenesis or even pre-fertilization on the beach. So these spontaneous mutations can occur in both male and female. but there is a skewing for how frequently they occur and what the risk factors are. So for example, they tend to be a little bit more common in sperm DNA than in egg DNA. And part of the reason for that is probably obvious. The male reproductive system is mostly external. Therefore, it's more prone to absorbing UV light. Ovaries are tucked nicely into the abdominal cavity and somewhat protected. There's also a difference in the number of those methylated cytosines that's present in the sperm DNA than in the egg DNA. Because there's more methyl Cs, there's more of a chance that this type of modification would occur. Now, guys, there's also something else to consider, and that's that sperm turns over pretty rapidly. And anytime something is moving at a high rate of speed, proofreading errors do happen, even if they're very, very infrequent. The typical male will make about 1,500 sperm per second. That's about 130 million sperm per day. When you're making that many cells and going through that much DNA replication, there is incorporation of errors. And in fact, the error rate tends to be about 1 every 1 to 5 billion bases. You've got 3 billion bases in your genome. So every time that you're making a sperm, you're introducing 1 to 10-ish changes just randomly, unpredictably because of the proofreading systems. Now, sperm are very fast made, but they're also very short-lived. So it's only around 6 hours. So if you're out in the sunlight on spring break in South Beach and you're going to wait 10 or 12 hours before doing anything that would result in fertilization, you've probably made new sperm. And this really is meant to highlight that it's not something that we have any control over. It's not something that's done right or wrong. It's just something that is. Because things are turned over so quickly and sperm only lasts for about 6 hours, these mutations don't really accumulate over the lifetime. You don't have a sperm that's from 10 years ago. So the things that happened 10 years ago aren't going to be recaptured in the DNA of the sperm. It's a very short process. That's for the males. Now, for females, it's a little bit different because, of course, females are born with every egg that they will ever have. There's no replacements. They don't turn over. So any mutation that does occur in the DNA of an egg will remain throughout the lifetime of the woman. And that does allow for the possibility of cumulative alterations over lifetime. Okay. Now, in the recent years, a lot of these genome comparison studies have started to come out where the sequences of parents and their children are being scrutinized in very granular detail. And what's being found is that there's often about a dozen, or normally about a dozen, just de novo spontaneous DNA changes that occur at random sites in the genome between generations. And so this is, we think, part of how the evolutionary process, if you will, is continuing on, in that there is some scrambling or some modification of DNA just naturally occurring from generation to generation. Often it's actually at the expense of those cytosines, or the C bases. And so I said there's 4 bases in the genome. If everything was equal, you would expect each to be present at about 25%. And that's actually not true. The number of the number of C's and G's that are present in the genome are a little bit less proportionally than A's and T's. And probably this is the reason, is that more of those alterations convert C's into T's, and some of those changes do indeed cause genetic mutations. So why is this important for epilepsy genetics? Well, as I mentioned, these spontaneous de novo events do represent a leading cause for new genetic forms of epilepsy in a family. And this would be something that has not been seen in the family. It's a patient who doesn't have any family history for that type of an epilepsy or a condition. And the genetics would indicate that they would be the first person or the founder for that particular trait or phenotype. It is very possible that this could be passed on from generation to generation, and a genetic counselor or genomic sequencing analysis might be required to really identify what the risk of transgenerational transfer would be. These types of changes can affect any gene. That makes them more difficult to diagnose. This is being better addressed with genome sequencing procedures and methodologies that have really taken off over the last roughly 10 years. And so it's not as large of an obstacle as it used to be, but it still is often difficult to identify what the causal mutation was that led to the epilepsy condition. And as I mentioned, we know some genes are more prone to others to these types of de novo changes. And it's probably, again, the way that the DNA is structured in the gene or the genome of the sperm cell or the egg cell is structured. Some are a little bit more exposed. And so we do have some more common events that take place rather than just being random anywhere with equal chance. So as I mentioned, you could very well be the first person in your family to have one of these spontaneous DNA changes. And because of that, it is possible that it could be passed on to your children. It's complicated to some extent because it depends on the type of the DNA mutation. And a genetic counselor and genetic testing would probably be the most straightforward way to assessing the likelihood that something where you were the first individual in your family to show this particular feature, whether it would be passed on to your children. Okay, why don't we see if there's any questions about the first part of today's presentation before we jump into what makes boys and girls different?
We do have a couple of questions. I'm not sure. One of them is, are protein deficiencies common in gene mutations associated with epilepsy?
So that is a very good question, and it depends on the type of mutation. So in some cases, and you've probably heard the nonsense mutation, if you have a genetic report and you have a mutation where you have a X after a position, so like R294X, any type of that sort of nomenclature would suggest that there is a protein deficiency. If you have a different type of mutation where it doesn't cause a truncation or elimination of the protein, it would change one of those amino acids in the protein itself. And so that would be something that would fall under the category of missense mutation. And the protein is almost certainly going to be present in those conditions, but it's changed in a way that hinders its functionality. And that hindrance can be dramatic. In some cases, the protein itself is still expressed, but it has no functionality. Or in other cases, it may be hypofunctional, or it has a little bit of its function, but not enough to maintain the brain circuitries within their normal homeostatic balance. So that's a great question, and it does depend on the type of mutation and the mechanisms that I just reviewed on how those mutations occur are equally likely to promote either type. So I hope that answered that question.
Yes. The rest of the questions are more about, I think, the second half of the presentation.
Okay.
So
shall we go on and talk about boys and girls and their differences?
Perfect.
All right, X chromosome inactivation. And this one is going to be geared more towards the ladies in the audience, but it does highlight a couple of factors that can explain why conditions are also more preferentially or more predominantly seen in males and not females. And I'll try to touch on both equivalently as I go through this section. So, you know, what is one of the biggest genetic differences between men and women? And of course, the biggest difference is because males have a Y chromosome and only one X, and females have two X chromosomes. My wife loves to tell me that this means women are twice as good as men because they have two X chromosomes, yet we have but one. And unfortunately, there's a fallacy in that, because in any cell of any lady in the audience, there's only one active X chromosome. Yes, you inherited one from your mother and one from your father, but only one of those two remains active in any cells. So we're equal. Boys have one, girls have one. Okay. Key point. The one remains active and the other one remains silenced or inactivated. Now, usually in women, this is a 50-50 mix. So half of the cells in any given female will express the X chromosome that was inherited from her father, and the other half, excuse me, would express the X chromosome that was inherited from her mother. Okay, but sometimes those patterns aren't 50-50, and they can be skewed towards one or the other X chromosome. And so if you can imagine that there might be differences on the genes on the X chromosome inherited from mom or the X chromosome inherited from dad, it's very, very possible that the patterns were different between mom and dad on their X chromosomes. So if you silenced mostly your father's X chromosome and left mostly your mother's X chromosome active, then those traits that were encoded on her X chromosome would be expressed in you more preferentially than those that came from your father. And skewing happens normally. It's just a distribution of luck. And it can influence how things on the X chromosome are expressed or not expressed in any individual. And you guys have seen this. I want to show a little video here that illustrates it very, very clearly. I'm going to see if I can run this. The individual who's going to narrate this is Dr. Huda Jagbi. She was nominated for a Nobel Prize this year and received a Gardner Award, Canadian Gardner Award, about 10 years ago for her work in neurodevelopmental conditions. And this is a video that was provided by the Howard Hughes Medical Institute. If anyone's interested, there's a whole complimentary set of these animations. They're freely downloadable and freely usable. You can get them just by Googling Howard Hughes Medical Institute and X chromosome inactivation. Anyways, let me just play this video, and then when it's done, I can review a little bit more.
And these cells start off in a female with two X chromosomes. The cell divides, and the embryo is getting slightly bigger, and still each cell has both X chromosomes active. But in early embryogenesis, each cell will inactivate one of its X's, and one cell will remain with the paternal X as active, while the other one, the maternal one. And now this process will happen at random, and you'll have almost half of the cells with the maternal X active, half with the paternal X. The embryo will continue to grow, cells divide, all the descendants of that cell, all the descendants will keep the same X active. And you've seen this in calico cats, right? The coat color gene is on the X chromosome, and that's precisely why these cats look like this, a mosaic appearance, because of the process of X chromosome inactivation.
I will venture that probably everybody has seen a calico cat at some point, and calico cats are always female, unless they have a trisomy of the X chromosome, but they're always female. And the black coat color gene came from one parent on one X chromosome that the cat inherited, and the orange coat color came from the other parent on that X chromosome. Now, has anyone ever seen a calico cat that has exactly the same coat pattern as another? I haven't, and believe me, I've looked. Same's true for any two women in the audience or anywhere out anywhere, is their pattern of X inactivation, or if you would, what would be the equivalent of their calico cat coat color, is unique. And that's dictated by the patchiness of, if we use the cat as the example again, the black versus the orange coat color. Now, have you seen calico cats that were more black with little patches of orange and also orange with little patches of black? And that you do see, and you do see it quite often. That's X chromosome skewing. So if we then go back into our clinical conditions, what if we had a gene that resided on the X chromosome that was only affected, say, from the father's side? And two women or sisters inherited that same X chromosome. One had X chromosome inactivation that silenced 80%, 90% of that X chromosome that was received from her father, while the other sister silenced, say, 50% or 60% that came from the mother. They have the same X chromosome inherited from the parent, but because of the skewing differences, one would express more throughout the body of the X chromosome that contained the dysfunctional gene as opposed to her sister. And so that can influence the severity of a genetic condition. Now, guys, we have only one X chromosome. We're always the worst case scenario because there is no mosaic patterning that's present in guys. And so that's where some of this difference in sex, tolerance, or effectiveness can originate. If it is a product that's on the X chromosome, there's no backup in males. Whereas in females, it's even possible that with extreme skewing, the inherited X chromosome that had the mutant gene may be completely silenced, and therefore the individual would still genotypically or still have the gene, but phenotypically there's nothing that happens because all of the cells are expressing the normal product. And as I mentioned, we're now very much appreciating that a lot of epilepsy conditions are caused by mutations of genes that reside on the X chromosome. Just a few examples here are the Fragile X mental retardation syndrome genes, FMR1 and FMR2, Protocadherin 19, CDKL5, the Rett syndrome gene, MECP2, ARX, HNRNPH2 is another X-linked gene that's involved in a pediatric seizure phenotype. The PIG-A gene, IQSEC2, the IDS2 gene that's responsible for Hunter syndrome. There's a strong database, and it's growing, unfortunately, of new genes that reside on the X chromosome that are being found regularly. As I mentioned, the effectiveness of males and females with these mutations can vary quite dramatically, and that's because of the single X chromosome inherited by males versus the mosaic pattern that's present in females. And as I mentioned, X chromosome skewing can very significantly affect the severity of the condition that the individual will eventually develop. And as a sort of an anecdote, I can give an extreme condition of this. Sandy mentioned in my introduction that I work on Rett syndrome, and Rett syndrome is a neurodevelopmental condition that affects almost always girls. And seizures are arguably the issue that most severely affects the quality of life of these patients. And I know twins, and they're monozygotic twins, they're identical twins, and each has exactly the same mutation of the Rett syndrome gene MECP2. One twin inactivated 99.9% of her X chromosome that contained the mutant MECP2 allele. Last year, she graduated from Columbia University with honors. Her identical twin inactivated 99.9% of the normal X chromosome and expresses in almost all of her cells the mutant, and she's very severely affected by the condition. And they are identical twins genetically, but obviously very far from identical phenotypically. And so this skewing can have pronounced consequences with respect to whether an individual will even perhaps show the condition or have a mild form or perhaps even be very severely affected. And again, this is where genetic testing would be very helpful to identify if an individual did have a skewing of the X chromosome and what likelihood there would be of something being transmitted to offspring. Okay. Any questions there that came up during the discussion of boys and girls?
Yes, we did actually.
Okay.
One question is, what about in the case of a frame shift mutation?
On the X chromosome, the answer would be if it were an X-linked gene, the same properties would be in play if only one of the two X chromosomes were affected by that frame shift event. If you're asking the question about whether a frame shift mutation would cause a protein to become absent, it depends on where the frame shift occurs. Most frame shifts do cause the loss of the protein itself, but not all. And so I'd have to have more of the specific circumstances where that frame shift occurred to really be able to predict whether some protein would remain or whether it would all be degraded and removed from the cell.
Okay, thank you. Another one is, in the 1980s, they said epilepsy was not genetic.
Yeah.
Later, they started stating it remained not genetic, but the tendency to develop it may be genetic.
Yeah.
To what degree has this changed over the decades, and if so, why?
Well, you've shown that question highlights the projection of what's happened. When I was a graduate student in the 1990s, I was working in one of the laboratories that was sort of pioneering some of this genetic epilepsy research. And we didn't really think that a lot of the epilepsies had a genetic cause. We thought most of it was exposure or early life infections. If you fast forward about 40 years, what we thought was in play certainly is. Environmental exposure and infections absolutely are sufficient in some cases to cause epilepsies. But what we thought wasn't going to be the case, where genetics would be a major determinant of one's susceptibility, has clearly proven to be the case. And I think that what I highlighted in one of the earlier slides is that we're now looking at 2/3 to 3/4 of the idiopathic epilepsies are probably genetic. And with genome sequencing of more and more patients, those rarer mutations or polymorphisms that collectively change one's seizure threshold are being identified. And so I would say that in 2020, I think that's generally accepted that genetics, and I'm about to talk about epigenetics and lifestyles, are major predicating factors for where one's sensitivity will be to developing seizures. It may not be by itself enough to cause a seizure. It may require something else in addition to it, but it sets where the threshold is for how likely it will be for an individual to have another event that would cause you to go over that, if you will, threshold to develop a seizure. That's a really good question, because it also highlights that the research efforts are required in order for us to learn what's right when what we originally thought probably wasn't.
Okay, thank you. And there's one more here. What are the chances of carrying epilepsy into later generations? If one parent passes the genetic mutation onto one of their children,
right,
and the other children are not tested, what are the chances that that gets passed?
So my first comment on that is it's an excellent question that's hard to address. It's sort of a topic of itself, and it depends on what type of the epilepsy mutation is involved. There's dominant genetic mutations and then recessive genetic mutations. And so if this were talking about a dominant mutation, it would mean that only one copy of the affected gene, either from mom or dad, would be enough to promote the condition in the child. If it's recessive, then both the mother and the father would have to contribute a altered gene to the child. And so if it's a dominant mutation, then the unaffected children have very little likelihood of transmitting something to their own children. But if it's a recessive mutation, then you don't know. And this has been termed previously as carriers, where I mean, I can use my thinning hairline as an example that came from my mother, but my mother had a perfectly good head of hair. She was a carrier. And because it's an X chromosome and I don't have a second X chromosome to counter it, I developed the receding hairline phenotype. And the same is true for epilepsy. If you have a recessive condition, it can be in the genes, but it's not manifesting because the other allele or the other gene that's present from your other parent compensates for it. And it's only when both of those genes, the one inherited from mom and the one inherited from dad, each have functional impairment, that the child or the individual would develop the condition. So I hope that sort of helped address the question. It's a great question. It's just a little bit complicated because it's context dependent.
Right, right. We have one more question. If someone has genetic testing done and it is determined that they have a mutation in the gene such as TSC2, is it possible for that mutation to be benign and that they wouldn't further be diagnosed with tuberous sclerosis?
Yes. So again, you guys, excellent questions. So you remember I mentioned the difference between polymorphisms and mutations. I'm not the only one that occasionally is a little cavalier with using the term mutation and polymorphism. So what you first described would be a change that's benign. That would probably in a purist's world not be called a mutation because it's benign, and it would just simply be a polymorphism. You may also hear this called as a single nucleotide polymorphism or a SNP. And those happen all the time. They're just changes. They do change the gene a little bit, but it's not in a way that adversely affects the function. So if it is truly benign, then it's probably not something that would be transmitted or developed later. Now, what if it's something that isn't truly benign, but that it's anticipatory? And so then it would be a mutation, but it's a mutation that requires time for whatever its consequence will be to manifest. And unfortunately, we do have a lot of examples of that as well. In that case, then it could develop later, and it could also be transmitted transgenerationally because something else needs to happen in order for the consequence to be fully realized. I'll talk about what some of that other stuff could be in the next section, but in some cases, and tuberous sclerosis is one of those examples. Sometimes the mutations in individuals are tolerated very well and nothing happens, but the same mutation in a different individual can cause tubers and epilepsy phenotypes. And it's not always easy to predict which will be the case from a person-to-person basis. We know statistically what roughly the number would be, but statistics don't actually look at an individual. They look at groups of individuals. And so I would probably suggest, again, a geneticist, a genetic counselor, or somebody that has a little bit more information about the specific mutation and the history, the natural history, and they could probably give you a better answer or a more accurate answer for what the likelihood of that event developing into something later on in life or transgenerationally.
Perfect. And that's all the questions we have for now. So
That's a great lead in though, because it gets to one of the points of the next section that I want to cover. And that's your genetics are set. What you get from your mom and dad or what happens spontaneously, that's not really going to change. Yes, you can get little minor changes. It's almost never in all of your cells unless it happens very early in embryogenesis. But your genetics are just that. Excuse me, they're fairly set. But we know that the environment, your life experiences, they can sculpt how the genes that you inherit function. And so that's the sort of the next section that I wanted to delve into and talk about this idea of epigenetics. And that's how life events and the environment can influence how your genes function. And so just as a quick review, I'll try not to get into too much granularity with this, but epigenetics just simply refers to processes that regulate the structure of your genomic DNA within the nucleus. And I've got cartoons here that show a whole bunch of different material. The take-home point is really shown here, and I'll show it on my next slide as well, is that the DNA is this blue strand here. And in the nucleus of your cell, it wraps around this other structure. It's a histone structure. And that's normal. Now, if it's wrapped too tightly, then all of the coding that's present on the DNA is going to be squished up against this protein. And that's really hard for those genes to be accessed by things that allow them to be expressed. So if your epigenetic structure is tight or condensed, then it tends to cause the genes to be incapable of being expressed. And the flip side is also true. If the DNA is loose and it's not as tightly wrapped around this histone thing, then it's much easier to be accessed by those gene reading or transcriptional complexes. And it's much easier for those DNA sequences to be read and turned into protein. And this is a dynamic balance, and it's influenced by day-to-day activities, and it's also influenced by all sorts of different things. I'll review a couple of them in a moment. There's even some evidence that these epigenetic changes may actually be inheritable across generations. And so epigenetics really is where the concepts of nature and nurture intersect, and they influence the genetics that we receive from our parents. And so this sort of shows the same thing in a different way. The DNA itself wraps around this histone protein as shown here. The genes again are present on the DNA, and the epigenetics will dictate how tightly it is associated with that histone protein. And we know that it's a dynamic process, and it's also critical during early embryonic development. And so I posted another little video that Dr. Jagvi will narrate again, and it really shows this process very clearly, and it brings into play a couple of the modifications that we talked about earlier. But the take-home point, I think it'll be clear, but we'll play it and then see what happens.
So here's the gene being transcribed, and next to it you see acetylated histones, nice open chromatin, and now you catch these little Methyl-Cytosines. Methyl groups is appearing, and here comes the protein. It binds and will attract this protein complex along with the enzymes, the deacetylases, and now acetyl groups are gone, and histones become tight, chromatin closes, no transcription factors can come through, and the gene is silent.
So highlighted there was how the genes associated with that histone area would be silenced. And so what you saw was some event took place, something in the environment or a cue or something happened that caused a change of the DNA to take place that allowed this other thing to bind to it and restructure that region of the genome. And that restructuring in this case would silence the genes that were associated there. No change whatsoever in the genomic sequence occurred. The gene structure is exactly intact. It's just how functional it is has changed. The opposite is also true. You can do the reverse and make areas of the genome that are less active, more active, using a similar process. And so that's sort of the model of how this epigenetic or nature and nurture influences would be transcribed onto our genomic DNA and regulate when, how, where, and to what degree those genes are being expressed. And this allows cues from your own experiences, as well as cues that you would get from the environment, to actually be incorporated into how much our gene expression control, our gene expression control systems. The level of this genetic regulation will occur potentially throughout the entire genome, but we also know that there are some genes that are more subject to this epigenetic influence than others. Now, I'll give you a great example of this. Probably, I'm going to go out on a limb here and say that the majority of us probably have had chicken pox. Well, chicken pox isn't a great thing to have active in your body throughout your life. It's a virus, it's a pox virus. When you're infected, it actually integrates into the genome, into your genome. So why isn't it active? Well, those epigenetic processes that Dr. Jagby just highlighted silence it. It gets methylated. That chromosome gets condensed at the region where that virus has integrated into the genome, and it's silent. So when you have chickenpox as a child, you'll have, a week, maybe 2 weeks where it's not so good, and then it gets shut off until later in life, when some often stress, but perhaps other thing happens that changes that epigenetic silencing and allows the pox virus to reactivate. And that's called shingles. And so that's a beautiful example of how this epigenetics can really influence expression, with the pox virus being the target of the epigenetic modification. And then the reactivation would occur because of something else, stress certainly being sufficient, but probably other factors as well, that allow it to be re-expressed for a short period of time. Fortunately, it is generally then silenced again, but certainly is unpleasant, if not infuriating, during the time when it is active again. Moving on a little bit, some of the sites that are regulated by epigenetics are permanently altered throughout your lifetime. They don't change once that genetic or epigenetic code is laid down. It's permanent, but other sites can be dynamically influenced. If you just look at different people, it's clear that many genomic sites, the same site in the genome, can be quite different in its epigenetic pattern between individuals. And this can even be if their DNA sequences are identical. And so this is, again, where I'll give a little bit of a short story where this is probably the best appreciated. A colleague of mine named Art Petronas at the Center for Addiction and Mental Health in Toronto took a look at the epigenetic patterns in 2 young women who were identical twins. Again, these are monozygotic women. They grew up in the same household. They were raised under virtually the same conditions. But one of the twins, her lifestyle was a little bit different than her sister's. One twin was more conservative, worked in a bank, drank in moderation, had one boyfriend who was her partner for, pretty much all of her adult life, was pretty good with saving money. Her identical twin was a war correspondent who jumped out of airplanes into firefights, drank like a fish, could not keep a boyfriend, and didn't have a cent to her name. Now, they're identical twins. So when Art looked at their epigenetic pattern, he found that they were completely different. And if you just looked at their epigenetic patterns, you wouldn't have thought that they were related at all. Now, the question is, did the epigenetic patterns cause the behavioral differences, or was it the behavioral differences that then caused the epigenetic pattern differences? And that we don't know, but it just illustrates that correlationally, you can have quite different epigenetic patterns that correlate with quite different behavioral tendencies and or life experiences. And certainly the same is true for all of us at certain regions of the genome, including those that are associated with stress responsiveness. We can have different patterns that cause different responses to the same stimulus. And in the case of epilepsy, we believe that that can also be a significant contributing factor to how severe or even if the epilepsies will develop and manifest. So if you put it all together and you look at our genetic sequences and then how nature and the environment play against one another, we have our genetics, and I've listed this here as our inherited genetic burden and our life experiences, and that's our acquired epigenetic modifications. And both of those can equally contribute to what phenotypes or what behaviors or what personalities we will have in our early life, middle life, and later life. Now, in some cases, the inherited genetic burden can be quite strong, and it can overcome or be predominant over anything that the environment or life experience would supply. And then the alternative to that is that the inherited genetic burden can be overcome by epigenetic modifications. And one example of this could be post-traumatic stress disorder, where an event causes epigenetic modifications that are sufficient to promote a phenotype that arises somewhat independently, or certainly not dramatically prevented by the genetics that we received from our parents. And so all of us fall into one or perhaps more of these different camps, and the different features that we exhibit are the culmination of what you see here, in some cases being significant from the environment and through epigenetics, and in other cases being more influenced by the genetic code itself that we received from our parents. So if you put all of this together, and this gets back to one of the earlier questions, it allows us to model how these different factors, what we inherited from our parents and then the environment, establish where we are with respect to our own susceptibility to developing epilepsies or seizures. And so if you just look at this graph, each of the points or the spots on this panel represent individuals. And it's just color coded just for ease for appreciation. There's two different black bars that are on the graph, and they represent two different types of individual. And the bar here, the lower bar, would be the threshold for somebody to exhibit a seizure, but it requires a stimulus. It requires something else, maybe the flashing light or the auditory sound on a railway, or something that's a clear cue that is sufficient to cause a seizure in these individuals, but in its absence, they wouldn't display a spontaneous seizure. Some individuals, their genetics and epigenetics are so far below that line that they're not going to develop a seizure even in the presence of the stimulus. But there are some individuals whose genetics and epigenetics, and those would be the spots here that are above this first line, who are prone or at risk or have the thresholds that would allow that stimulus to promote a seizure. Now, there's also another group of individual, and that's shown by the higher bar here, where their genetics and their epigenetics collectively put their threshold above a set point for spontaneous activities. And so this would be, for example, a channelopathy, or one of the neurodevelopmental conditions associated with an epileptic encephalopathy, where the genetics itself, and it is sufficient to be a single gene, has put their set point on this seizure threshold graph in a position that's above what is necessary for spontaneous epileptic activity to emerge. And so we are all on this graph somewhere. And where we are is a combination of all of those different features that I discussed throughout tonight's presentation. The take home here is deciphering what causes one to have a predisposition for seizures may someday be able to help guide the most effective treatment strategies. But because we're all individual, deciphering that information has to be done on an individual basis. It doesn't do any good to say this works for John and Mary, and therefore it should work for Paula. That's not necessarily true, because Paula may be completely different in how her system placed her on this graph at some point, and the treatment that would be required to properly manage her condition would be unique to her. And we refer to that as either personalized medicine or more recently precision medicine. And that's sort of the goal for research labs like my own, is to try to better appreciate how one can convey precision medicine into individuals rather than prescribing a drug that works in 75% of the people, but you don't know whether you're one of those 75, or of the 75, you're one of the 50% that would have some severe side effects. Okay, so that's another stop point. I've already gone way over with my timing, so I think it's probably a good point just to end. I would be happy to address questions. And the last part of my talk, I was just going to review gene therapy and precision genome editing. I'd be happy to spend a few minutes for those who would like to stick around to address a couple of developments in this field. I'd also be happy to answer questions and just go from there. I apologize for being long-winded, but obviously this is an area that I enjoy discussing, and I hope that there's been some information that was helpful for you guys that was covered tonight.
Definitely, definitely. We do have a couple more questions.
Sure.
Would autoimmune issues or inflammations be considered as genetic?
Oh, that is another great question.
These seem to be areas being looked at for more difficult seizure disorders such as Byers and Norris.
Absolutely correct. So here's the deal. We don't fully know what in all cases causes an autoimmune condition, nor do we know fully what causes an inflammatory reaction. And probably as we learn more, we're appreciating that one's genetics influence the magnitude of both of those responses. So from a simplistic view, one would not necessarily say that an autoimmune condition or an autoimmune cause for an epilepsy condition is genetic. But when you start turning the rocks over and you look a little bit more closely, you start to realize that the reason that there is the autoimmune condition to begin with can very much be because of a genetic undertone. Same with neuroinflammation. Some people respond with large immune responses to the same challenge that other individuals have very little or no neuroimmune responsiveness. Why? And as we learn more and more, we learn that the genetic background influences, and perhaps the epigenetics as well, the magnitude at which that inflammatory response will proceed. So I think as of today, the best way to answer that question is controversial, but if you ask me it in 5 years, I would predict that the new information that's available between now and then will almost certainly say that there is a genetic component that's at play.
Perfect, thank you. This is the last question we have. Do you have any sense of how or why there seems to be such a predominance of epilepsy being comorbid to the autism spectrum disorder and intellectual disability, and how genetics and epigenetics come into play for this common combination?
So thanks for that question. This is actually one of the areas of focus of my own research program at the Krembil Research Institute. And the answer is it's probably associated with the impaired homeostatic balance in the neural systems that are a consequence of whatever the genetic cause for the autism spectrum disorder, or the other neurodevelopmental condition. Now, this doesn't refer to channelopathies, or it doesn't refer to some of the other epileptic encephalopathies where there can be structural changes in the brain. This would be where no structural changes are readily evident, but yet the patient still can display hyperexcitable networks. And so the question is, why has the excitatory system become dominant over the inhibitory system in the brain? And the answer is we don't know, but there's a lot of speculation that it has to do with the homeostatic balance between the two systems being quite different from one another in the consequence of those mutations. And those mutations, seemingly, even though they're not related to one another, have a convergence on how they impact the circuitry systems of the brain. And the net result is that there's too much entrainment. The excitatory system becomes too rhythmic upon itself, and it loses the inhibitory tone. And that's why often GABAergic drugs, or drugs that block the excitatory system, would be the first pharmacological tries at managing the epilepsy in the patients. How does that happen cellularly is a hot topic of debate, and there's no definitive answers that are out there at this moment. But at least from the circuitry perspective, what is the culmination of many of those different, seemingly unrelated genes that are affected in those neurodevelopmental conditions is the allowance for the excitatory system to become too rhythmic, and that allows it to entrain, and that's what allows it to reach the seizure threshold and cause the seizures. I hope that helped to answer that question. That's a great question.
Perfect. One more question just came in. Whenever you discover that a certain gene mutation triggers seizures, what treatments are available for those people other than anticonvulsants?
And this is a question where I wish I had a better answer. We don't have a lot of options. Our toolbox has quite a number of drugs in it. None of them are perfect. Some of them in some patients work pretty well. A lot of them in many patients work, but they're associated with some pretty significant side effects. And in some cases, none of them work at all. The ketogenic diet, you guys may be familiar. Actually, Ottawa has one of the better ketogenic diet platforms and clinics present in Canada. It can be remarkable for some of these types of epilepsies. It's challenging, though, for a family to maintain control of a ketogenic diet, particularly in a child. But that certainly would be one option that I would encourage someone to discuss if the drugs are not being well tolerated, or for other reasons, unfortunately, financial can become one of the mitigating factors, aren't the first line or the preferred first line of treatment. The next two things that I was going to try to discuss tonight are things that are possible, that are coming in the pipeline, but they're not available in the clinic yet. So I'm afraid that as of this moment, lifestyle changes, diet, at the extreme end, it would be the ketogenic diet, or the pharmacotherapies are pretty much the tools that are in the toolbox. There is neurosurgical. I've mentioned that here, the last bullet point on this final slide. In some cases, the neurosurgical resection of the epileptic tissue can be the cure. And in some individuals, their epilepsy has formed in a way that makes it very amenable to surgical treatment. That's generally not the case for neurodevelopmental conditions. It's more the case for a spontaneous epilepsy that's occurred particularly later in life and due to some other event, be it a tumor, a vascular malformation, perhaps an infection, perhaps a trauma. But that certainly is something else that could be considered. The fear, I guess, or the concerns with the neurosurgery are very well warranted. But when you actually look at the numbers, the patients who benefit significantly from the neurosurgical treatments, particularly those who have a high likelihood of surgical success, is actually much better than those who are managed pharmacologically. And so that's something else that you could put in the toolbox for consideration, although it's something that's not applicable necessarily to all patients.
Mm-hmm. Sorry, I just did find one more question.
Yes.
The SCN1A epilepsy, Dravet, is
Channel
already
apathy.
testing gene therapy
Yes, it
in
is.
select patients. Is
Yeah.
anything happening in Canada with other ion channel epilepsies like SCN8A?
Yeah, so the answer to that is no, and the degree to which those clinical trials have started are still in their infancy. If you'd like to stick around for a couple of minutes, I can sort of go through why that is. But yes, with a couple of actually types of Dravet syndromes, there are some companies that are pioneering gene therapy programs. Rett syndrome also has, I think it's now been approved, a clinical trial for gene therapy. They're very, very early days, and the technology is showing glimmers of hope, but it's probably not yet far enough along to be sufficient, at least in a large group of patients, to be a cure. And whether it would show more benefit or less benefit from some of the more conventional strategies, I think still remains to be determined.
Perfect.
I'm not aware of any place in Canada that is currently conducting any of those types of gene therapies.
Okay. Well, I'm not even sure what time it is. I'm pretty sure we're pretty.
I'm showing 5:30, I think. Yeah, 5:30. So I was afraid that I would be a little long-winded. Why don't we just call it a day?
Sure.
And if there's interest in having me back to go over some of these, or if you'd like suggestions for somebody who could also touch on these experimental strategies, I'd be happy to either give it a go myself or suggest somebody who would probably have a bit more expertise than I in those specific areas. It's certainly a fascinating field. It's evolving quickly, but I will say that for most cases, it's still at the early experimental stage.
Okay. Wow. Well, thank you so much for joining us today.
Oh, thank you for having me. It's
it.
been my pleasure. And oops, I'm trying to give you back the screen here. And there.
And I found it most informative. Thank you so much. And we look forward to our next webinar.
And if other questions do come in where I can be helpful, you have all my contact information. Feel free to send them on, and I'll do my best to answer them.
Excellent. Thank you so much.
If anyone out there is a U.S. expat or has dual citizenship, let me wish you a happy Thanksgiving and enjoy the festivities in a safe way. And to all, happy holidays upcoming, and please may everyone stay safe and secure. Let's weather this storm together and get it behind us so that we can go back to doing the things that make life life.
Absolutely. Thank you so much.
You guys have a great evening. Thank you.
Take care.